Thanks, Min, and thanks for the invitation. I really appreciate it. I want to talk today about some analyses that we've been doing with a very venerable data set that we've worked with for a long time, and that's good old Mod 13 16-day composite NDVI from MODIS. And I have to thank our NASA Stennis colleagues. They've worked very hard on this data set. They removed outliers and noise, and they've done a temporal smoothing, and then they follow that by a curve fitting and estimation of some things we call phenology parameters. Let me show you what I mean. This is an NDVI curve, annual curve, time across the bottom, and they find certain parameters. I want to concentrate on these 5 here in the middle. This is the 20%, the left 20% of the maximum, the left 80% of the maximum itself, and then post-maximum right 80% and right 20%. And we've used these phenology parameters. For each parameter, we can empirically observe 2 national scale maps, CONUS maps. The first one is the NDVI value when it reaches that particular point. The second is the day of year when it reaches that particular point. We're finding this very interesting. You can think of these as, we start to think of these as phenological milestones or completion thresholds. And even though they can only be calculated post hoc, they're arbitrary. And they're not trying to relate this to any kind of observable on the ground. They're not species specific. It's not bud burst nor leaf out. But instead, it's an actual property of the mixture of vegetation that's present, an emergent property of land surface phenology. And the good thing about this is you can paint these uniformly across national scales for analysis. So we think that these phenological milestones, as we've selected them, correspond roughly to start of spring, start of summer, the peak of greenness, start of fall, and start of winter. So we're trying to examine all seasons, not just start of spring. We have no particular fascination with start of spring at the expense of others. And of course, we can imagine that evergreens have less mixtures that predominate in evergreens have less prominent seasons, and of course crops are going to have human enforced seasons since they're placed out and then removed. So here's what I mean. This is looking in terms of greenness at these phenological thresholds. Here's spring greenness, and here's summer, and maximum greenness, and fall, and winter. But rather than looking at the NDVI value, the greenness, we can also look at these pairs, these partner maps, which are the day of year that we've reached the completion of these phenological thresholds or milestones. And so the purpose of what I'm doing is to try and figure out where each of these phenological milestones, which I'm suggesting are analogs to beginnings of seasons, are predictable across the CONUS, and also to rank and map the most important driver variables that might be influencing or controlling them. But before we do that, you can imagine that the most important drivers probably change depending on what vegetation type you're in. So we need some way to partition or segment the map into distinct phenological types, is what I'll say. And those of you who know me will not be surprised to hear that we use a multivariate geographic clustering procedure on this entire 12-year span of every 8 days of national data. And the object here is to group together cells that have a similar annual profile. And we created 50 of these phenological ecoregions. We like to call them phenoregions over this 12-year period. And you get a different map for every one of the years. And these maps move around geographically because, after all, there's a lot of interannual variation. And so these are not static, but instead are dynamic. And these phenoregion maps look like some of the best vegetation type maps you've ever seen because they are dynamically determined over such a long period of time, looking every 8 days. Okay, where do we get our season predictor variables? We're using a new improved version of the DayMet data set. It's from Oak Ridge National Lab. It's interpolated climatic data at 1 square kilometer resolution, and we merge these together, the tiles from DayMet for the CONUS for each one of our MODIS years 2000 to 2011. And we're harvesting 5 different variables from DayMet. The first is maximum daily temperature, and we accumulate that into a heat sum above 5 degrees since January 1. Similarly, we take the minimum daily temperature from DayMet into a cold sum below 5 degrees since January 1. And then we got precip that becomes a precip sum. We started that in March 1 because we're more interested in rainfall than snow. And then we use unmolested from DayMet, the day length and the vapor pressure deficit, which is a sort of an inverse surrogate of humidity. So we're not just using temperature, we're not just using heat sum, but rather we're looking at a broader set of predictive drivers. And we accumulate those top three right up to the day of year, which we have empirically observed that each of those phenological milestones were achieved. And now we're going to do multiple linear regression within each phenoregion. So in other words, we're using the 50 phenoregions as surrogates for vegetation type as a stratification for this regression analysis. Now, all these variables have different units of measurements, so we have to do some kind of a normalization, partial normalization. So we look at the full value of historical variability, the lowest to the highest, and then we convert the current value between 0 and 1. So we're really looking at the current value in relation to the observed full range of historical variation. Now you can just do the easiest thing, compare the 5 regression coefficients to see which of the influences rank the influences. So I'm going to show you a lot of results maps here. The main background on most of these maps is the R squared of the relationship, and it goes stoplight colors, dark green to yellow to red. And I'm going to show you a series of pie charts. And the pie charts that are superimposed on the map have one piece of pie for each of the 5 potential driver variables, and the idea is it'll show the proportional influence of each of these 5 predictors. And the pie chart sizes are also scaled by the R squared value. And we place the pies at the most representative location within each phenoregion. So again, like the phenoregions that are shifting around geographically, you'll see that the pies move around. I tried to pick mnemonic pie colors, so the red slice is heat sum, the blue slice is cold sum, the purple is precip sum, white is day length, and gray is vapor pressure deficit. So here's one of these maps. This is 2011, and this is the 20% left phenological milestone. So that's the analog to start of spring. You can see immediately that we got very high R squareds everywhere for this. Again, red is heat, blue is cold sum, purple is precip, white is day length, and the grays, there are not very many grays, but that's vapor pressure deficit. And at this kind of a scale, it's very difficult to see much of a pattern among the pies, but hang on. So here's the 12 years. This is 2000 at reading style here, and this is 2011. You can immediately see that spring is highly predictable almost everywhere, at least in terms of these phenological milestones. But you can't see the pies in this kind of presentation. So what I've done instead is I've taken the biggest piece of pie, and now I'm going to color the entire phenoregion by that largest piece of pie. One of the things we could see before, though, is that there's not any one of these 5 that are particularly dominant. So there's a risk in doing this. I'm only going to show you the winner and not the runners-up, so to speak. But when we do that, we get to see a sort of a consistent picture for the drivers of spring. Spring is controlled mostly by heat sum, the red color, but day length, the white, is also pretty prevalent. I think it's interesting that the 3 years that stand out to me as different, this one, and this one, I'll call your attention to these little numbers. These are the oceanic Niño indexes, and I've averaged them together for December, January, and February. And exclusively and exhaustively, the ones that are different here for the control factors for start of spring are the La Niña years. I think that's quite interesting. So what about summer? Summer is the hardest one to predict. We get the lowest R squareds, especially in the Pacific Northwest, the Desert Southwest, in the north central states, and in our southern Appalachians. And when we look at the drivers, it's an even more consistent pattern of heat sum as a driver everywhere, except day length seems to be controlling the southern Appalachians and the Ozarks. But we do get a smattering of cold sum and precip sum some of the time. Now I'm going to jump right to these max value maps for maximum greenness. And peak greenness has quite a different set. You start to begin to get some different flavors. I keep going here. Here's start of fall, and fall is not all that different from peak greenness, but day length is clearly becoming more prevalent, more influential. I see these very interesting double vertical sort of control structures through the central United States that I am interested in. And finally, here's winter. And it's quite clear that these are mostly white. So day length is predominating control in winter, although there's some cold sun controlled areas. Heat sum and occasionally precip dominate in some of the desert areas. Okay, well, I said a minute ago that there's not too much coherence among the pie charts when you just look at the whole nation, and maybe that's because it's kind of a mixed view due to different mixtures of vegetations and land use types. So we took the national land cover data set and we did a majority classification, that's 30 meters, up to the MODIS resolution, and we divided into 9 vegetation types: conifer, hardwood, mixed forest, crops, grass, shrub, urban, wetland, and non. I won't show you all those, but a very interesting thing happens, and let me show you that. It greatly increases the similarity of the pie charts within a vegetation type and within a particular season. So here's the 2000 map for spring that I showed you a minute ago. As soon as we drop everything out except for the conifers, you can see, if you stare at it as long as I have, that there are really only 3 different types of pie charts here. And this is a pretty interesting thing. The colors I've used to manually link these are just random, but I keep them the same across the types. Here's the same map of conifers in the spring for 2003, and although they're jumping around geographically, you can see it's still the 3 same kinds of pies. And in fact, if we look at all the years for conifer for spring, you see that these 3 types are the only ones we ever see across all 12 years. I find that this high fidelity of these 3 types is quite an interesting result, and I'm surprised by this consistency that appears in the phenological control through these years. And even if we switch to a different vegetation type, move from conifers to hardwoods, and move from spring to fall, we get a different 3 types, and in fact it's really 2. I think there's only 1 example of this 3rd type here. And so then they're not the same as the types for conifers in the spring. So I'm very interested that there's never seems to be more than 2 or 3 of these different families of proportions of control influence within each veg type and within each seasonal type. So quite an unexpected result. So initial conclusions, it's always a mix. There's not anywhere or any when that a single factor is dominating. I think that's interesting. Vapor pressure deficit of the 5 was the least important factor, but not completely absent. Day length control, I think, is almost a null hypothesis because day length changes with time anyway, and it's sort of been fixed evolutionarily. There's a progression we've seen across the year. So heat sum is predominant in spring and summer, and then cold sum and precip, and then finally day length. It becomes more important later. We got some INSO, La Nina influences, and I'll speed up here. I'm fascinated by this. I don't think it's a fixed, locked relationship between a particular set of phenoregions and a particular NLCD veg type because the number of phenoregions, the number of pies varies. And I'll skip to the end now. I know that was fast, but you can see my slides if you'd like to go in a more calm way at this initial URL. And there's 3 ways of looking at these in these next 3 web pages, and you don't have to look at tiny maps. You can click and you get a big version. And finally, this 4th one here is our phenoregions page, phenoregions clearing paths page. So if there's time, I'd be glad to answer questions. Otherwise, thanks for your attention.